The next organelle to discuss is the nucleus. The nucleus is the largest organelle and contains the genetic library of blueprints for synthesis of nearly all cellular proteins. It responds to signals that dictate the kind and amount of proteins that need to be synthesized. Most cells are uninucleate or have only one nucleus. Some cells are multinucleate or have more than one nucleus. For example, skeletal muscle cells. Mature red blood cells are anucleate or have no nucleus. The nucleus has three main structures: the nuclear envelope or membrane, the nucleolus, and chromatin. The nuclear envelope is a double membrane barrier that encloses the nucleus. The outer layer is continuous with a rough ER, and like the rough ER, is covered with ribosomes. The inner layer, called the nuclear lamina, is a network mesh of proteins that maintain nuclear shape and act as scaffolding for DNA. The nuclear envelope encloses the nucleoplasm, a jelly-like fluid in which other nuclear elements are suspended. The membrane has pores that allow substances to pass into and out of the nucleus. The nuclear pore is guarded by the nuclear pore complex, which regulates transport of specific large molecules. The nucleoli, singular, is nucleolus. Nucleoli are dark-staining spherical bodies within the nucleus. They are involved in ribosomal RNA synthesis and ribosomal subunit assembly. Typically, there is one, sometimes two nucleoli per nucleus. Chromatin consists of 30% DNA, 60% globular histone proteins, and 10% RNA that is either newly formed or in the process of forming. Chromatin is arranged in fundamental units called nucleosomes, which consist of DNA wrapped around proteins called histones. Chemical alteration of histones have effect on DNA and therefore can help regulate gene expression. Prior to cell division, chromatin condenses to form chromosomes. So chromosomes are condensed chromatin. This condensed state helps protect fragile chromatin threads during cell division. This figure shows the increasing levels of structural complexity in chromatin, from the smallest to the most complex. The smallest structure is a DNA double helix with a diameter of approximately 2 nanometers. Histones package and regulate the DNA. A nucleosome consists of 8 histone proteins wrapped by 2 winds of DNA double helix with a diameter of about 10 nanometers. The DNA winds around each nucleosome. DNA clusters are attached by linker DNA. This electron micrograph shows the structure of chromatin with nucleosomes, which resembles a series of bits on a string. The next highest level of complexity is a tight helical fiber with a diameter of approximately 30 nanometers. The electron micrograph shows the next highest level of complexity, which is a looped domain structure with a diameter of 300 nanometers. The next highest level is a chromatid with a diameter of 700 nanometers. The next highest level is a chromosome. A chromosome in metaphase shown here consists of 2 sister chromatids.